Hi, welcome to Art of Academia, a weekly podcast featuring interviews with leading scientists and insider takes on life in academia, hosted by Komal and Madan, researchers from Cancer Science Institute, Singapore.
Yeah, so I was saying, I was also thinking of making this season 2, right? But at the same time, I don't feel like I have concrete ideas for big changes I want to make. So part of that was like, initially we were saying, right, we want to do more interviews.
Yeah.
And that is still true. But I also I kind of like the format we had. I don't want to change
but again,
it up.
I don't feel like seasons mean there has to be a major change.
Yeah, then what is the point of having new season?
This is like we are coming back kind of thing.
I guess. Okay, fine.
It's
I
more
guess
of that.
For that
it sort of drizzled down, but now we are like, oh, let's do it.
Sure.
If we do it or not is a different question. But the enthusiasm is there.
Sure, okay.
Yeah.
I thought we would make some changes. Like, for example, in my head, what would warrant a season 2 is like if we figure out video, for example. Like, if we're going to launch it with good video, that would make it like good. Okay, season 2, there's something
step
to
up.
there's something to look forward to. Or like you change up the format in a considerable way. Yeah. But I don't want to fuck with the format.
Yeah,
I think
like
it's
what would
working.
you even do differently in terms of the format?
So I just thought, I don't know, we'll become a bit more interview focused. But I think I thought about it and now I don't want to. Part of it was like there was this pod on the not overthinking and Taimur was just like bashing people who do interviews in the sense that everybody does it, right?
Yeah.
And I think it's true. Like actually everybody...
I mean, it has its own value for sure. You can now know so much about them and
yes.
things will come up which you would normally never think of.
Correct. But
the mix of both is still okay with me.
True. So I feel like what we do... No, in my head I was like, we should do more interviews and reduce our one-on-one stuff. But I think
at this point there is no reducing.
Yeah.
There's just like doing something. Yeah. Fair enough. I get you.
So hence I feel like let's hold off on the season 2.
No, I'm still on board with that.
Yes, I know. It's for us, obviously.
I do feel excited about...
Yeah. So that's what. Do you want to use that as, new season, fresh start?
Yeah.
Okay, fair. Sure.
Yeah.
I can keep video for season 3.
Sounds good.
Okay, then. New season. What are we doing? Starting with the same format. What's up with life?
What is up with life? I can't even remember when was the last. So what to update our listeners?
Should we do a recap? Should there be like a explanation as to why there was such a long break?
Sure.
I feel like maybe...
No, we owe that to the listeners.
Yes.
Okay, go for it.
Okay, so I guess what happened was... Well, firstly, you were in between jobs. So you got fairly busy.
Yeah.
But also I was writing my thesis.
Of course.
And especially closer to the end, I got too much. So I guess that was really the main
seriously,
reason.
Yeah. Because I remember discussing this in one of the previous episodes where both of us were like, this is happening for me, this is happening for you.
Yeah.
Let's see how things go and then we can revamp.
Yes.
Yeah.
So now that I guess you're settled into your new job. And
someone has submitted their thesis.
My thesis.
Yeah.
Yeah, so hence we are back.
Yeah.
Although that being said, I feel like there's more interruptions coming. Yeah.
Why do you say that?
Or like defense, for example.
Sure.
But yeah, I think apart from that,
I think it is still better than like the writing phase. I am sort of at least a bit more chill now.
Yeah. I don't know why I'm so scared to make a public promise. You know,
it's just fine. Nobody cares.
Yeah, nobody cares. But you would care, right? I don't know.
But like both of us know that we want to do this, and if it is not happening, it's not our fault. Like in a way, it's like real things which are coming in between. Because even for me, for that matter, okay, we will come to things anyways. And then there was travel as well.
Yeah.
For me, like multiple times. For you as well, a few times, I guess.
Yeah. So what else? What has been up with life? You might want me to go first?
Go for it. Yeah.
I guess if you have to really catch up, that's a couple of months worth of updates.
I think this will be like a long catch up episode. That's fine.
Yeah, let's see. What are big updates? Yeah, I guess I've also been like traveling a lot, right? We mentioned. I mean, it's not that much, but to me that felt quite disruptive. So after I submitted my thesis, went back to India once, then came back. Then there was this research seminar thing, and then I went back to India.
I never talked
again.
about it. How was it? Did I ask you? I can't remember.
Did you ask me? I don't remember also. It was okay. It was not... I think I put...
Oh, I did ask you. You said Ashok was not there and overall, like, my lab people asked questions, but apart from that, it was chill.
Yeah. There was a lot... So there was some conference going on. Okay, wait. Context again. Research seminar is supposed to be like this big... I don't know if you felt it, but it's supposed to be like this big meeting, right? Where you show your work and then you I guess everybody sees it, so there is something to it outside your own lab.
For sure, yes.
And in my lab especially,
oh my god, your lab is a little too much, to be fair.
And they take it very seriously, right? So it's like
every small thing, man.
Multiple rounds of practice, fixing the tiniest details in the slides, everything. So I was coming from that place of like, this is the norm in the lab. It's a decent amount of effort, much more than I normally would. But I felt it was not worth it because there was like nobody to sort of like give me some critical feedback. Like I was expecting Ashok to come up with like his usual killer
all of
question.
This is useless.
Yeah, exactly, right? And I
and then you're ready to like sort of defend.
Yeah, I was really prepared for that, but that didn't happen.
Yeah. That does happen like for several presentations, I feel.
I guess so.
Yeah, that adrenaline rushes there and then you just want to... And it's okay, I guess.
Yeah, it's okay. I mean, there's nothing to be said. It just felt a slightly anticlimactic.
Yeah, I feel it.
Anyway, going back to the previous story. So basically, I have stopped doing work. I had stopped doing work for a very long time. Thesis writing, traveling, research seminar, coming back. And then now I went back again, right? And then I was chilling.
Yeah.
So I guess now my general state is that, okay, let's get to work and do something.
That has been the vibe for such a long time for me as well. Every time.
I think you do yours. I want to hear your version of this.
Okay. Anything else?
No, you do this.
Yeah. Basically the same thing about travels, because I travel even more than you do. I would even go for like 4 or 5 days, just like include a weekend and just stay with my partner or like family. And my mode of working is a bit different though. Like I try to keep doing some minimal work and in the background, even when I am home or like I'm traveling. Not like proper travels, of course. Like if we're going to Bali or something, I won't be working.
Yeah, hopefully. I hope not. Pull out your laptop. I have my defense. I'm not planning to do that.
No, actually, you know what? Okay, I will come to grants in a bit. But there was a deadline which I was planning to apply for, and I was like, shall I take my laptop and just do this thing if I'm not able to do it before then?
Okay, fair enough.
Anyways, but the point is, because I travel so frequently and I can work remotely, doing more computational work, I like to keep that momentum.
Yeah.
I like to just be like, okay, while I was here, I had all these blah blah targets or like these few explorations, analysis I wanted to do. I did it. And now like I have this more chill time at home where of course I'm spending time with whoever and some background work, which I will now pick up after I come back. Obviously, it doesn't like stay with that momentum all the time, but it's nice to have something in the background. But every time I would come back, it's still like, again, I just came back from there, settling in the home and like all the chores and all of that. And then again, going to lab and just settling back to your life in a different country again. So that obviously... And then small things would happen and you're just like, my friends visited.
Yes.
So that was another like almost a week of just going out again and again, doing random things. And I just had this feeling like deep down, I want to work. I just want to get something done.
Seriously, man.
Yeah, I'm tired of like this kind of on and off, not like, full on mode.
Yeah, which is interesting because I don't think either of us are very grind set people, right?
Yeah.
So I guess if we are feeling it, that is symptomatic of something. Which is, I mean, I don't know what else to say apart from that's just interesting.
This is slightly tangent but still in connection with what we are talking. I feel like I have several modes of work. So when there is like no deadline on me, normally of course nobody likes deadlines.
Yeah.
So I would be doing more explorative work, chilling, like my life is like, get up in the morning, I feel like playing the ukulele, I'll play the ukulele and then go to work till late. Like basically it's all very flexible.
Yeah.
And the research and the things I'm doing is also super exploratory. Reading, finding something excited, just following that. And nothing is super conclusive or, robust at that point. But then somehow some deadline would come, be it a meeting or like something I need to send to people or whatever. Then I get into that mode of, enough of playing around. I have played around enough. Now it's time to sort of make this into a piece which other people can understand. Make this into something which is, you can be sure of and like be conclusive of.
Yeah.
What stresses me out the most is like when I have to do something for someone. I think I've told you this before.
Yeah, you have.
Like I'm completely happy working. I enjoy working. But when it is like somebody has asked me to do this and give this to them, I will procrastinate on it like anything. And it stresses me out because now I know like this thing is there that I need to send, which won't take super long, but I'm still doing this random ass thing.
Yeah.
And it just piles on. The longer it goes, the longer I procrastinate. So but it is something I really need to work on. But I just need to be a more people's person, and collaborations are obviously the way to go. So
Yeah.
I need to definitely improve on this mindset.
Yeah, I mean, I totally get it. I feel, I mean, like somehow I'm not in this position, right? Because I guess I just do my own project. Not that the people are not working with
Yeah.
me. But if I was in your place, I think I would have this exact same problem.
Yeah.
So fair enough. How do you do that though? How do you get better at it?
So one thing was, of course, if I also struggle with admin things I was telling you. So maybe I was thinking I can just put some time on tasks like this, which are not so explorative, not so research-wise exciting to me, but then like maybe an hour or two in a day, and be like, okay, stop on whatever you were doing, let's just do this.
Yeah.
So it just gives me like sort of a foundation on the time, because normally it's always like I start on something and I get lost in it, and then there's not enough time, or I just don't feel like doing the other thing.
Yeah.
That's one way. Apart from that, like practically, how can I improve this? I'm not sure. Like mentally also, I'm aware of this, man. Like there are things I need to send to
maybe
people.
That is step one.
As
right?
well,
just
yeah.
acknowledging that it's a problem.
Like it's not, I like to help people, I like to collaborate, but somehow this thing it's just strange. And then like I will start avoiding them as well, because obviously I haven't responded to whatever they had asked me to. And eventually it's favorable to me, like my name goes in those papers. So this is weird.
Yeah. Yeah, I get it. Well, I guess the biggest part is just starting, right? Because once you start, maybe then you get like locked in, and then it's easy to continue.
Yeah.
Yeah, I don't think I have anything very insightful to say about this. I think these kind of problems I have not solved in life.
Yeah.
But you better have a thesis by next episode then.
Yeah, I mean, I think just the two things that we discussed are doable, for a start, and then I'll see how things go from there. Yeah.
I feel like the setting time aside thing, I feel like that would work for you.
Yeah. Not for you.
No.
Yeah, I think it would, yeah.
Yeah. Okay.
Cool. What else? I was telling you, there are some like grants coming up. So there was one in A-Star which I wanted to apply, but the deadline is like in a week's time, 31st October. So initially I thought maybe I will just do it because I have stuff, but I felt it was too rushed. Given we are also traveling, and it's just not ready. But there is another one which is quite new, and like I really found it quite exciting. It's called forget the name, Discovery Catalyst Fund, something like that. But the point is, it is for postdocs, like research scientist level 2, 3, whatever categories they have, and it is to sort of increase collaboration across labs. So what the how they are gonna go about it is, there is a pitch and match soon, where everybody, all the participants, like postdocs, will go and do a 3 minutes elevator pitch. And then you just match with other postdocs from different labs there. So you could either have a team already of 2 people, or you could go there and like pitch your idea, and then you can collaborate or find a collaborator right there. And then the overall idea is to start something completely new. You will be given like a year or 2 to work on it, decent funding, and then from there, if something preliminary comes out, like something interesting comes out, then you can use that to apply for bigger fundings.
Nice.
Yeah, so overall, I just like enjoyed the idea of this pitch and match, where maybe now I can find somebody who's keen on this and like just increase my network. And of course, it's a competition, but in general, like I like the idea of this. Yeah, if something like this happened where both of us were there, I would have like, just loved to do this together. Now you have the money. You have a real project, and then it's like a bit more serious, more official, and then like 100%. You can just work on it
and this,
more seriously.
This feels nice compared to like traditional grants, because I guess even if you don't get the grant, you get something out of it. You get to like see all
other
these
people's
other people's
ideas.
pictures. Maybe you talk to somebody. Yeah. Normally you just write and send something in and it gets rejected, I guess.
Yeah. That's the norm.
Yeah.
And even apart from that, like this is at the level of postdocs. So normally it's like either you apply as an individual young postdoc and then use your PI as a sponsor or something, or it's like really PIs, and then you are a tiny person in between. But this really feels like, okay, both of postdocs are sort of at the same level, and
so there's no PI in the loop.
Yeah, I don't think so.
Oh, that's interesting.
I do not think so. Like eventually, of course, you would need their name and the lab, because that's where the funding would come. But it seems like it's really the PI would be us. We'll see how that goes. Yeah,
So
need to think of ideas. It is in November.
Okay. Have some
8th
time.
November or something. So
maybe not that much.
Yeah. I have like signed up anyways. So you could pitch, or you can't even, like, you can choose to not pitch even. So you can just attend, and then in case, somebody's pitch is nice, or like you can still go talk to people. I plan on pitching, of course, like that increases exposure and it increases connectivity. I have some ideas from the ongoing work that I have been doing, but I don't want it to be super similar to what I do already, because conflict of interest and like this, my PI is not attached to this institute, blah. But there are like maybe 1 or 2 things which I could use from there, not directly related, and then explore it, because most of the research I'm doing is still things which are like resource or description from all these sequencing data sets. So now more exciting would be to pick a few of those insights and then see if validate them, or like find ideas which could be exciting. And apart from that, of course, if somebody else has something nice, we can think on those things, and eventually obviously need to get the grant to get anything going.
Sure.
But it's a nice exercise.
Yeah, I feel so too.
Yeah.
I think even without the grant, this would be worth it.
Yeah.
Any other life updates? We're at 20 minutes.
Life updates. Trying to be financially more stable.
Wow. Okay. What does that mean?
It means like I have, of course, done like investment and stuff. I plan to invest in a house with my father. So maybe that would happen later this year or like next year. That's a big move. So this is like a flat right across my parents' flat.
Oh.
Yeah.
That's
it's
very
right
wholesome.
opposite. Exactly. That's the whole point. Like if it was somewhere random, I would not have thought about this much investment and like all of this effort. Long-term future goals are like I can just come back, stay there. My parents would also feel like, okay, this girl lives here. So
okay, very cool, exciting stuff. Very adult stuff.
Yeah.
Cool. Shall we start then?
Yeah, let's do it.
Okay, so I was basically reading these 2 Substack essays. I think one of them I shared with you, which was called "Why Haven't Biologists Cured Cancer?" Yeah, cancer researcher. What are we doing, right? And it's a nice essay. I'll link it in the thing. People can read about it. And I don't want to summarize the whole thing. But basically, Ruxandra Teslo, I don't know if that's a guy or a girl, probably a girl. She talks about why progress in biology has been slow, right? This is something people complain about, cancer especially. We've been working on this problem for, I guess, close to a century. Made some progress, but really not satisfying given how much money we're pumping into it.
Yeah.
And she goes through like a few reasons as to why this might be. So the first obvious one is biology is just very complicated, right? Like you just have to concede at that. There's a lot of variability if you compare that with like physics or something.
Yeah.
She also considers a few interesting points, which is how all the smart people go off to do like physics, computer science, and only the dumb people go to biology. So maybe that's why we're stuck.
Lol.
But she's like, no, that's not the case. Biologists are also smart. So she goes through a few of these cases, and what she essentially lands at, the main problem, or one of the bigger problems, is slow feedback loops, having very long feedback loops. And I actually, from this article, I found another one by a guy called Stephen Molina. I think he's a software biologist, if that makes sense. He's just a software guy who now works on biology. And I think they're building software, I think, for bio people. I'm not entirely sure what he does. But anyway, so he wrote a longer piece that was more focused on feedback loops also. So these 2 was really my inspiration. So we'll just get into what that means, right? So let's talk about what a fast feedback loop would be. So this is something where you do an action, right? And then you get your response or the outcome very quickly, almost instantaneously. And the fastest this you can imagine is, I guess, sports, right? If you're like learning to kick a goal in football, you kick it wrong, it's not going to be a goal, right? You instantly know that something went wrong, and then you can course correct.
Yeah.
So you can change by like kicking slightly to the left, slightly to the right, little bit more force, little bit less force, blah blah blah. And this would be an example of a very very fast feedback loop between your action and the outcome. And I guess the whole premise of this whole idea is that fast feedback loops can be very magical. The funny thing is, when I was reading up for this podcast, right?
Yeah.
We actually already talked about this in at least 2 episodes. So the first one was in wet lab versus dry lab.
Yeah.
In fact, there is this literal segment where we complain about wet lab has very slow feedback loops, right?
Yeah.
Which is very relatable.
Yeah.
Anyway, so that's how they link it. So the core problem of biology is that the feedback loops are very long, and part of that is because just life, cells have to divide, and that's something we haven't engineered to make faster, right? So you just have to wait. And that just scales up. Like once you go from cells to mice, that takes even longer.
Yeah.
You go to humans, and then now that's taking decades.
Yeah.
So that's like the main problem. And I think in the past, even us, when we talked about it in the wet lab versus dry lab, we sort of conceded that yes, that's the case.
This is what it is, and we deal with it. Yeah.
Yeah. And then in fact in our episode we were like, that's why bioinfo is good. Right,
which
because we can do dry lab stuff, which is actually very fast feedback loops.
Yeah.
Before we proceed, I do want to make this point also, that because we talked about this aspect in very old episode, which was applying software principles to research. So here the idea is that one of the reason people get so hooked to programming, or I guess one of the reasons that software has become such a big success with so much progress very rapidly is very short feedback loops. Like you can write code, run it, and you instantly know if something has gone wrong. If there's a bug, it doesn't run. And I get a lot of people have actually written about that, right? And even in that, we talked about how one of the big inventions in the software industry was to make these even more shorter, like make the cycles even more shorter, so you're getting your results very quickly and you do multiple iterations. And I guess there's more examples. I think a very, very, very prominent one, but a slightly different one, is AI. I don't know if you've ever seen this video so this was before ChatGPT existed even. This was basically a video showing how AI could beat like the best Dota players, for example. Did we talk about this at some point?
AlphaGo.
AlphaGo even better, I guess. But I don't know the details of AlphaGo, so I'll stick to the Dota example. Basically, they built this program to play Dota, which is like normally a very complicated game. And I mean, human players, they learn by... And so you can't really learn it by rules. You have to learn it by playing because there's so much complexity. You do random things. And sometimes some things work and some things don't work, right? So the way you learn is by just playing a lot. And then eventually you get better. And the thing with AlphaGo, the Dota version, was that it could play thousands of games at the same time against itself. So even though I guess a game could not go faster or whatever, it could like scale up in a different way,
right,
and get a lot more feedback, right? And eventually, of course, it ended up beating the best players of the game because they are limited with how many they can play. They can only play one game at a time and only so many games in a day. So I guess just many examples of even if like everything is very super complicated, super inefficient, whatever, if you have like fast feedback loops, you can make it work. So that's like the broad idea. So that's I guess what I wanted to talk about. But before I go into like specific ones, do you have any reaction?
Reaction. Again, like as I mentioned, I was thinking step by step and I have forgotten some of it. But I think one point about dry lab versus wet lab, in the previous conversation, we just accepted that yes, dry lab has much faster feedback loops. You can do things faster and then you can get results and you can move on quickly, which is not true in the case of wet lab. But now that I do full-on computation biology, that is true for sure. I can just start in the morning. Within a few hours, I am already there with some decent results. Of course, finding it if it is true or not is like, it takes more time to do all those corrections and stuff. But then, at least for me, I do have that longing now for validations or like, some experimental clues, hints into if whatever I'm seeing is real or not, or like how would this now actually play out in a cell. So just like sort of trying to correct on the conclusions we made in that previous episode. That even it is more driven by the interest of the person. Like, of course, there will be people who would be super happy with this super fast feedback loop now. You just do your thing, but then I still have this extra longing for wet lab component.
Yes.
The other thing
wait, before you move on, I want to react to that.
Yeah.
It's funny because actually, how you said about the episode is also how I remember it, but I actually went and re-listened.
Okay.
And we do have a segment in the end where we both were like, yes, TriLab has this component, but it is actually missing this final thing of like some sort of validation. Like if you want to do some intervention, you can't, right? You need to do experiments for that.
Exactly.
And I think so you were making that point, and at that point I was like, yes, I agree in principle, but to me that was not a very didn't sink in, I guess.
I see.
But I think lately it has sunk in.
Right.
So I am feeling this more, and hence this problem of like, okay, we can't give up. So what can we do about it?
I see. The other thing I was gonna say is, of course, you will talk about how to shorten these loops in your system and what are the ways you are thinking about it. Like now I'm trying to think about this from the perspective of what I do, and my work right now has been super translational, where I am sitting in meetings with clinicians. I'm sitting in meetings with all these people who are basically doctors, and they always talk about patient level things. So now I'm thinking, if I want to shorten feedback loops in the sort of questions that we are asking, then how do we go about it? So I think maybe once you start, and this is also food for thought for later, I think, because the questions that people ask in the lab are more about, of course, how would this drug work? Clinicians don't really care much about how things work. They just want things to work.
Yeah.
If it works, they will. If it is safe, it is doing miracles, we will treat, because that's just their goal. But to actually do that is not easy. As a biologist, you would obviously always think about all the sorts of things that could be happening. For them, treating somebody with a drug, and if the tumor shrinks, is all that matters. But now, how is this drug really working? What else can it do? What is the mechanism and things like that are questions that I am more keen on. But again, very complex systems. So how do you now shorten the feedback in such sort of area?
Yeah.
And lastly, just like a fun thing, which we had, I think, also discussed in the previous episode, was about non-science things, like the creative things that we do. There again, we see faster feedback loops. Be your writing, be my like art, or like anything of that sort. It's just like just doing something, keeping me like reaching it to the finish line. It teaches us so much.
Yeah.
It teaches us now to be able to now restart again, and now how this new feedback would work. Whereas in a research project, a lot of times it's just like you are doing like step by step, but you have never reached the finish line. Of course, the finish line is also a bit unclear in research, but it's so hard to reach that finish line to be able to accept that, yes, I have done this loop. Now let's start something new, or like let's expand this and things like that.
Yeah, for sure. A very silly way. So this idea, right? We probably have talked about this, but this problem of a research project being too long, and exactly that of, is there a way we can like go through the cycle faster, right? Just go through a few projects, few shitty projects, till you get like a good idea of how this is done. I did have this thought, but I feel like it's just so hard to implement in real life, because first, I guess research is never, even if it's a fun time pass project, it's never like such a small unit of time that it's like insignificant.
Publishing is like not just like, okay, you did
these
few experiments, you saw this, and then blah.
Exactly.
You wouldn't want to do it by yourself anyways.
Yeah, so you can't, if it's too sort of not detailed or not enough work, that's never going to see any publication. Forget about even a shitty journal, right? So the second option then, I guess, is to make it sort of more fun. And then just publish that on your own. Like, don't look for publications, maybe you just put that out on your own, and perhaps other people will like still give you some comments, and maybe that's good enough. So I guess that was sort of where I settled on this, and I do have a few ideas for just fun projects that you can do on the side. Not very concrete, but we are actually doing one. I think, I don't know if
I know about
you
it.
know about this, but I don't know if I want to talk about it now.
Maybe a bit later, but where I would just continue on where you left off. I think given the wet lab, like just doing wet lab, of course, whatever you just said is very true. It's difficult to sort of make things and then make proper conclusions or like proper findings and then publish. But for dry lab, given that in the situation that I am, most of the hard work of collecting tissues and sequencing and all of those painful things have been done. So sort of like my not drive really, because of course the science is also the major driving force, but sort of in a way, like plan is to be able to use this data and then do some fast publications and then get that kick, just that feeling, how that feels, how that helps you, and then take a pause and be like, okay, how can I be doing this like for super long, or like now how do I want to change what I have done in the past year or so, and then take it from there. But in the shorter period of time, I am planning on getting these loops completed, a few of them completed, and then just experience that and just be able to do that, and we'll see how that goes.
Yeah, I mean, it sounds like a solid plan. What my not really I wouldn't say concern. So the reason I said that, part of that is the wet lab thing, but also why I wanted to keep it that's really, really super, super short is because if you spend, I guess, a lot of time on side projects, there is this thing as your main project, which eventually gets affected, right? And I feel like I always sort of underestimate that side projects do take away from your ability to focus on like the main thing. So I guess for me, it's also a little bit about like balancing that, right? Because the more you start expanding these side projects, the less room there is for your sort of the main thing that you're doing. And this is like such a obvious but like harsh truth of reality, I guess.
I guess this is also one of the ways to make the feedback loop shorter, to focus on one or two things instead of 5-6 things, because I do that. Like I will get some finding, and then I would just get excited, and then I would get into that rabbit hole. Whereas all of this analysis is looking at me. Woman, you just left me.
But the thing is, like with the main project, you can't, even if you do that solo only thing, maybe for
not solo even. Like I'm not even saying this is the only thing, because obviously that would get boring. That's the thing. It's risky as well. You don't know, if it will reach fruition and stuff like that. But what I'm saying is like maybe, because the thing is, I think for both of us, starting something new and like getting interesting ideas is quite exciting, and then it's easy to get like in that flow, and then forgetting about
the other stuff,
other things. So this being a bit more mindful of that could help in keeping the feedback loops shorter for particular things, for those particular more focused projects. Because even apart from research, we also do like random non-research things, and that also, anyways. I think that's fair. Like, I guess, yes, probably in the purest sense of feedback loops, completing sort of different things might not count, but there is, I guess, a certain momentum that you build, right? Even if the stuff you learned from something is not transferable, the kick you get out of it, I guess, that is transferable. That is true.
So fair enough.
Fair, yeah.
Okay, so I guess maybe we'll move on to how we can actually shorten these feedback loops. This one is very obviously specific to me. I want it, I don't know how, like eventually I want more people to be able to like think about this and like, I guess, contribute. I don't know, maybe part of that is just, I guess, making the case so that more people think about it. But yeah, where we're starting is just my stuff. Seems like a reasonable place. Actually, I think I already told you about these. I feel like we talked about this at some point.
Our listeners don't know.
Sure. Okay, so let's start with cloning because why not? I found out because recently I was doing a bunch of cloning, and I was just looking things up, and I realized that there are like actually a few ways in which you can make things go faster. And then I'll give you one sort of final magic bullet that makes everything I say redundant in the end.
Do you want to quickly describe what's cloning?
Oh, do we have to? Okay.
Very briefly, like just simple layman.
That's a good point. Okay, so cloning, I guess a very common biology experiment starts with what does this gene do or what does this protein do? And being a stupid biologist, the best way to figure that out is to either remove that gene or at some other times make a lot of that gene in a sort of different system, right? And then you just see what happens. I guess that's the very naive way to explain that. But there's a few problems, right? You can't just directly get a gene to go into a certain thing, blah, blah, blah. So you need a few systems, mainly vectors. This is just like bits of DNA that code for the gene that you want to make.
Yeah.
And making this is what cloning is.
Right.
Yeah. I hope that made sense.
To me, yes. I don't know
Okay,
listeners.
okay.
Yeah.
Yeah, so basically most experiments start with a cloning step, right? And that being said, I also got a recent appreciation for how much better this is right now compared to
Okay.
in the past, you know. I
From
just
all your history blogs that you were reading, yeah.
I went into a little bit of like rabbit hole digging. And my God, people in the past, they suffered, man. and really, I feel like some gratitude for all the engineering that has been done.
I mean, that is why like the smallest things that we said smallest now were such big discoveries back then.
Exactly.
Like even that Venki Krishnan?
Wait, what's his first name?
Venki Ramakrishnan.
Venki Ramakrishnan.
Yeah.
Sounds not right, but okay. Okay.
Yeah, but even in his book, which I did not finish, but
I finished.
Oh, let's go! Finishing feedback loops.
Yes.
But basically, just the technology development also was a major theme of the book, right?
Yeah.
The protein structure, crystals, how to see them, the microscopy, how it was developing. If you have to like sort of do that same experiment of looking at ribosome structures today, it's so easy.
Yeah,
all of those tools are there, but like because it was so damn hard at that time, it became like a Nature or whatever paper back in the day.
Yeah, and like more specifically, like apart from enabling stuff that was just simply not possible, it's also much faster and much easier now. So that how you have to say it, like give that disclaimer. So
like
thank God.
I heard like not even 20-30 years ago, no, not 20, maybe that woman was 20 years ago a researcher, but basically one of my senior postdocs in the previous lab, she told me like there were these Western blot gels, the ones we load now, in small tanks, you just load your thing. There was like, they were huge that you would climb on a ladder.
Oh.
Like they were super long, like physically, like human size. So you like climb up and then you load your thing on the top and then you wait for I don't know how long because it's such a huge thing. And then it would like separate.
No way, this
I don't know
if
it was Western blot or like electrophoresis, something of similar.
Why does
like
it sound like she made it up to fool you guys? Like these youngsters will believe anything.
No, she was too sweet to be like fooling us.
Okay. Wow, that is so hard to imagine. Okay, yeah. So that being said, we've made a lot of progress, but things can still be better. So I'll just give you a few examples. One thing that I found, so I basically found this on the AdGene protocol, AdGene database, website. What am I saying? AdGene website. Because they do a lot of transformations, right?
Yeah.
AdGene is a company that sells plasmids. So they have to do this a lot. So one thing they do to reduce the time it takes for transformation is they use this special kit where the bacteria, they don't need to be heat shocked to be transformed. You can just mix them and then that's just done. So that saves like a nice 30 minutes.
Yeah.
And then I looked it up. The kit is commercially available.
Yeah.
Same price almost. So I guess no reason to not change, So that was like a one very simple change where that just cuts some time off, shaves some time off.
Yeah. Are the bacteria different? Like how is this happening without the heat shock?
Yeah, exactly. I don't know.
No.
That's the one. That's what I meant by I was trying to figure out how that works.
Right.
But I think it's some buffer that makes it open up. Right, and then there's a wash step, I guess, to close it again.
I see.
But I don't know.
Yeah.
But okay, maybe more interesting one. This is a new strain of bacteria called Mach1. Mach1. I think Mach is like a unit of speed. Like these. MACH.
Oh.
Like
not heard of
did we go for that Tom Cruise movie together?
No, I was not.
You were
Top
not
Gun.
there. Top Gun.
No.
Okay. So like if you're going at super fast speeds, they say things like Mach 9, Mach 10. I think it's a pun on that. There's a Mach 1 competent cell that basically grows super fast compared to other commercial bacteria. I don't know if it works, but if it's true, for example. So normally when you do transformation, how does that work? You do your transformation this evening and then you plate your cells on a plate. Next day morning you see colonies. You kind of wait again till the end of the day and then you outgrow these colonies in LB and then next day you can do mini prep and then you get your plasmid.
Takes like 3 days.
Yeah, 3 days. But because these things grow really fast, what you can do is like that morning, after your transformation and overnight incubation from your plate, once you get colonies, you can just take it and put it into your LB, and then before the end of the day, so basically in 4 hours, that thing has grown already to what you need. Instead of another overnight incubation, that just becomes 4 hours. So you can mini prep it on the same day and then send it off. So that shaves off a whole day of like just waiting.
Yeah.
Right? So I feel like that's great.
Have you tried it?
I spoke to tennis. It was not received very well. So that's the problem. And this one, fair enough, because the Mark 1, at least right now, they're selling that at a significant markup than the other competence cells. But the dumb thing is, we just need to buy it once.
And then you just keep growing it.
Exactly.
Yeah. Okay, you keep trying.
Yeah, I'm gonna keep trying. I'm not giving up yet. So this will. I feel I also didn't make the best possible pitch. So I need to like work on my pitching game, especially when it comes to buying reagents. Okay, so that was the second one. And there's a lot more. I don't know. I mean, I don't know how many of these you actually want to do, but I'll just maybe do one more example. Like also cloning.
Yeah.
So the last one was TA cloning. So TA cloning is just one method of cloning. So you have a gene of interest, right? You want to amplify that, but that's not enough. It needs to be carried onto a vector so that it can be propagated. And the traditional way of getting your gene of interest into a vector is restriction digestion, right? You cut it up, or cut both open, ligate, blah, blah, blah. Tedious, long. But TA cloning makes it faster because you can just like PCR amplify and get it in. And okay, I'll tell you about the engineering aspect of this, which is what makes it cool. So there is this topoisomerase, topo TA cloning. That's a variant of TA cloning. And what happens is, this company, they basically already have a linearized vector, a circular vector that has been cut open. So it's ready to receive your insert. But on either ends of the vector, there is a topoisomerase that is fused to the vector. It's physically... No, not to keep it open. Maybe that does keep it open, but not the main function. It's like linked to both ends of the DNA, right? And then now you take some gene, whatever you want. Maybe that's in the genome or it's in another plasmid, whatever. You just PCR it with the small thing that it has these A overhangs on both ends. So this vector also has A overhangs, right? So if you mix these together, they sort of hybridize. And this topoisomerase, by some magical property of it, basically seals that nick. And and then the enzyme fucks off, or I don't know, I guess if you transform it, you get new copies. So this is super great, right? And then there's just
the efficiency of this is much higher than the other existing
systems. And you don't have to do restriction digestion, right? Because they already ship you this linearized vector. You don't have to restriction digest your insert either. You just PCR it. Once it's ready, it's a super fast reaction. You just like mix both of these in a tube. 5 minutes, you keep room temperature, and then it's ready for transformation. And what blew me was like the iterative improvement on this as well. So like so many problems. Like initially, the kit, there was 50-50 chance that your insert goes in one direction or in the other direction, because both have A overhangs, right? You can't control it.
Right.
But they can make some modifications so that now it's like directional. So it now this ensures that all your colonies are in the right orientation instead of only half of them. So there's just like so many improvements. And even here, like we use the traditional cloning kit, and there are like clearly advanced ones. But fair enough. Like these again, they sell at a markup, and this is a consumable, right? You have to keep buying.
Every time you have to get your gene of interest.
Yeah, you have to buy their linearized vector. This is not something
the vector can be the same, no?
No, but it needs to be open and have the enzyme on it, right? So you have to buy the vector freshly. They don't give you a lot.
I see. Yeah, I mean, of course, these companies also...
I mean, they have to make money,
That's
right?
the whole point,
Yeah.
Okay, cool.
Anyway, so all of this, right? So many ways you can do different cloning. Or perhaps all of this entirely will become redundant with like gene synthesis, right? There's like so many companies now, like IDT is big, Twist, I think some Chinese company, super big. So their entire focus is you just give them a DNA sequence and they just make it for you, right? You forget about all this cloning PCR nonsense. You tell what you want and they'll put it in a plasmid and ship it to you. And the crazy thing is, with time, like where the technology is at right now, it's super fast. If it's just PCR product, you can get it in like 2 days.
I know, like a friend of mine in the UK lab, they used to outsource this all the time.
Right.
Like they just had a guy. I can't remember his name now, but he was also like good friends with her PI. And like whenever they wanted any new cloning done, they would just text him. Please, this is the
Please.
this is the
So good.
this is the insert.
Yeah.
And like that guy was also super enthusiastic, and then he would get back to you about like small details of what might go wrong, or like how would you want this thing to be, where do you want this restriction enzyme, blah blah, because these are all the considerations you have to have when you are cloning. And yeah, in a day or two, he would just ship it back to the lab. And they are never worried about any of that. Because again, this is a very cloning heavy lab. They are looking at a lot of genes, whatever they were doing, I can't remember anymore. But this guy was there.
Yeah. And yeah, so I guess I sort of knew about it in the background. But recently it came to my mind as to, or rather I realized that cost is comparable, right? And the turnaround time is fast.
Yeah.
So why the hell are we not switching?
Yeah.
This makes no sense to me, so now I'm like, trying to repitch it. I actually don't understand what people's hesitation is to using these services.
I guess like in a way people are not caring so much about, because the thing is, yes, it is spanned across 3 days time, the whole like at least for the bacterial transformation, blah blah blah, or like even cloning few days, but actual time is not super long.
Sure.
The amount of effort you would put in that one day would be an hour or so, or like 2 hours. I understand it is still more time than getting it for free, or not free, but like in short time. But you can be doing other things during that day. Does it really make it so much faster as you're claiming?
Well, I would say it probably does, right? Because in the 3 days example, we only talked about the transformation, but there is also like the design, ordering the primers, waiting for those to arrive,
and like the checks also, if the cut was happened or not.
Sometimes
and then
randomly things go.
Yeah.
But I want to highlight one quote that I wanted to say earlier from that feedback loop article.
Yeah.
It basically says, stop accepting slow cycles. Tech has many vices, but laziness and impatience are virtues that biology can and needs to learn from. I think this is so true, right? Just because, I can do it, and it's the same amount of time. Well, here I don't have to do it, and it's the same amount of time. So why not?
I'm completely on your side. I'm just like being the devil's advocate for like Dennis's sake, I guess.
Okay, I'll tell you what. Previously, I understood the hesitation because that would take time. Like the original company we spoke to, they were saying about a month, which is obviously long. I mean, if you do that yourself, maybe it's a week, right? But now it seems, at least maybe we haven't tried it, but it's promised that they deliver that like in very short time, like up 5 days if you tell them which vector also you want it in.
Maybe try that again then, at least like for one pilot order, see how that goes.
Yeah. Rachel's lab also uses that quite extensively. I think they do all gene synthesis. They don't like clone anything.
I mean, technically, like that is not the fun part at all. It's like just getting the system ready before you can actually do anything.
Yes.
But I guess for a lot of people, this counts as work, which it is. But it is not like impactful, useful, tangible work at all.
Yeah. Okay.
Yeah. Do you want to talk about like your change in model system?
Yeah, that was my main one, I guess. Okay, so the other, outside of cloning, big change, not the only change actually that I have made in my work, and this also sort of came serendipitously because we happened to be looking at this, not because I was planning to reduce feedback loops, which was going from human cancer cells to bacteria.
Right.
And what a ride, honestly. I just got on it, but I'm loving it. First of all, they are just so much easier to work with, right? Like, forget about TC hood. I can do that on my bench. I can probably spit in the culture and it's still fine. Whereas mammalian cells, it's like, infection, penicillin, what nonsense. Yeah, that is just the first thing. That they are just more robust, right?
Yeah, they're sturdy.
Yeah, they're freaking sturdy. And then the crazy thing is the doubling time, right? So for E. coli, it's 20 minutes. You can go from 1 bacteria to 2 bacteria in 20 minutes. Human cells take 24 hours, 24, sometimes longer.
Yeah.
And this is for one cycle, right? So if you think about it exponentially, this number changes insanely. So especially if you're doing big, large-scale experiments, like let's say chip, to go from a freezer stock, which is where you freeze your cells, right, store them, to have enough cells for experiment 1 week. And this is like not just throw and wait, right? You have to like take care of them, go and like give them some room. They don't like if it's too crowded, you have to move it, give them fresh media, all this nonsense. Bacteria, no. Overnight, just take it in a flask. Next day, the 50 ml full. They have grown to the and they're ready to go. That is crazy.
It is, for sure.
But the second one also, which I'm excited for, haven't done it, but it's coming up soon. So we talked about cloning, which is just like making the gene that you want to put into the cells. But how do you put it into the cells, right? For mammalian cells, very complicated. There's different ways to do it, but the lab commonly uses lentivirus. So first, you take your gene of interest. You have to clone that into a specific vector, and then you make virus. Now this virus carries this gene, right? And then you take the virus, you have to like filter that, purify that, all that nonsense, and then put that on the cells you want. And then now they have to go infect. They have
some
to
of the
integrate.
rare cells would accept them.
Yes, and there's some selection, right? All this happens. The whole thing takes like 2 weeks. And if that works. Bacteria, like we said, heat shock, right? Half an hour, they're ready to go. They're expressing. Next day, they're expressing already. So that's crazy.
Heat shock, man.
Yeah, heat shock, dude.
Shocking them, and they're like, just take this.
And they're like, yep, give me more. That's crazy. So I can't wait to like actually do the transformation. I haven't finished the cloning, so we haven't gotten there.
right.
Just thinking about it makes me excited. I guess.
That's great,
Yeah, just that. And also, this one is related to the first one. Just maintenance. I realize you don't actually have to maintain bacteria in culture, right? All you need is glycerol stock. If you want to do something tomorrow, start a culture tonight. Done.
Crazy, bro.
Yeah, tomorrow you do your experiment, you're done. Or like throw it off, don't you? No need to freeze them. One glycerol stock, you're set for life. That's insane.
That is really true. Like with my cells, I always used to struggle because like they would not freeze so properly, or like just don't have enough stocks, and then I have to be like very careful about what I'm using.
Yeah, and sometimes you thaw, they're not ready. You have to give them some time to get used to like being out of the thawing. You have to remember how many times they've been passaged, everything.
As if you did.
I never did, but I'm saying, right? Some people do that.
I know.
So I'm very excited for this bacteria shift, and already I'm like, is this sustainable? Like, can I keep the bacteria stuff going outside of just this one question that I'm like trying to answer right now?
I was just thinking about this as well. Like, this is again goes back to the sort of research you're excited about, the sort of questions you're interested in. If there's so many questions you can answer using this model, and it's just a matter of if they excite you. If they do, you keep on going till whenever.
To me, honestly, like of course there is the does this work in humans angle, right? But I don't think I've ever been that bothered by that.
Yeah, that's why I'm saying like it's more about the interest and excitement, because you would still get valuable insight from whatever you do. Most of the fundamental things have come from bacteria and yeast and things like that. And if that is the sort of vibe you want.
No, I mean, I like the fact that it's fast, right? But of course, you eventually want to tie that back. So if I envision, it's not like completely switching to bacteria, but having that as a side angle. So right now, why we shifted to bacteria is like, we see this phenomena in cancer cells, and we think it's important, but we don't know if it really is important. Right? There's this phenomena, but does it actually mean anything? So obviously I've had a few hypotheses, tried a few things, and nothing was really panning out. So one idea was, is this phenomena actually evolutionarily conserved? Right? Because if something is found across many species, or like in this case found in our furthest away relative, bacteria, that means that it's probably an important critical thing, because usually evolution is very efficient, gets rid of stuff that you don't need.
Correct.
So this would just be a nice way for us to like do some value addition in case we can't figure out does that actually have any implication? So I thought that's a cool angle, and that probably can be applied to few, obviously not to all discoveries, right?
Yeah. But that's what I was thinking. Like maybe it would be a fun exercise to think about the sort of questions and like, things that we can answer from the system. Even if you do it or not, it's a different thing. But just to be, aware of what are the things that we can do and how much of that is exciting and how much of that can eventually, of course, you can collaborate and you can obviously do your own things.
Yes.
Using other systems, but I think it's pretty cool. Yeah,
so we'll see where it goes. Already, like I mentioned, the peak of my fun times happening because everything is new, right? Like I'm reading up on new expression vectors. Things work differently. The bacterial genome is tiny. It's like 4 megabases. When I downloaded the genome, that file was 1 MB compared to like a 2 GB file that I'm used to getting. And so many like strange things that I don't expect, I guess.
I'm actually very curious on what are the questions we can answer using. Maybe I will read up a bit as well. Do they don't have like centrosomes and stuff, right? They don't, because they don't have chromosomes.
Do they have centrosomes? No, it's a circular. It's just like circular genome.
Yeah, actually. Yeah, then I don't
know.
No
telomeres also.
Yeah.
So wait, so they don't have centrosomes? Now you lost full interest.
No, I was thinking of like 2-3 things combined.
Wait, what is a centrosome?
Using centrosome.
You threw me off. Centromere, I've heard. What is centrosome? Organ now. Same as centromere?
I don't know if is it same? Centromere? No, centromere is the DNA structure. Centrosome is the organelle itself.
Protein that
attaches to the spindle assembly. All those spindles that attach to the kinetochore of the chromosome.
Okay.
And then they help in division of the cell. It's just that I'm thinking about this for some questions that I was dealing with, and then I was like, oh, they don't even have centrosomes. Yeah.
But like, for example, RNA polymerase, only 1, not 3.
It definitely like reduces the complexity by I don't know how many folds. Yeah. Like during several discussions in the previous lab, I think Ashok, of course, being Ashok, he loved this system because
bacteria,
yeast, bacteria, like these very simple systems which were there in his time, right? Early, like, I don't know, 2000s, 1990s, a lot of things being discovered through basic systems. And he would many times like point out to, if this is the question you want to answer, maybe you want to go to a system like yeast, where things are much simpler and then you can get some conclusive answers than be like, this protein is redundant, or there are like, these are the pathways which also do the same thing. Now you see something clearly, you get to know what this protein is doing, and then you come back to your own system where obviously the complexity is higher, but you have more confidence in what you observed in this one model system.
Interesting.
So for sure it's interesting. Again, depends on person's excitement and priorities and what they really like.
I guess a lot of aversion to that might be coming from, actually I don't know, what is the aversion to going to simpler systems?
It's mostly, I think that it is difficult to translate these things. If
it doesn't feel important.
In a way, like it still is, because if you can find something fundamental, it's super important. But again, how much can it explain to a cancer biologist? That is the field that we are in. Most of the clinicians would just be like, I do not give a shit. This does not tell me anything about what would happen, because even mice experiments don't tell you really what's going to happen in a human, and if that is what you care about, not useful. That's all. But I think there must be a group of people who are like hardcore.
Yeah, of course.
There is obviously there are obviously. So I think it's pretty fun.
Yeah, and we haven't given up on yeast either. So there's a very good chance that whatever we see doesn't hold up in bacteria, right? Probably highly likely that it does not. So then the plan is to fall back to yeast, which is like closer. But for that one, we have a collaborator. So that one I think I won't do.
Yeah.
We will outsource
Sounds
that.
good.
I guess outsourcing is the ultimate
for sure
feedback loop.
That is what I do need to jot down my notes from the Night Science podcast that I've been listening to. But one of the biggest thing that most of the people talk about, 2 things. One is interdisciplinarity. So them being able to talk to other people and like connect ideas from different fields. And the other one is collaboration and having good people to think with, talk with, work with, and then be a team and just like do things together as opposed to being super competitive. I think that is definitely the way to go.
So there's something to share. Why don't we just briefly talk about that book, the Gene Machine? And I remember that because of what you just ended your sentence with, which was like to say talking to more people, right? And that was like the one thing that stood out in the book from me, from his story, of this guy, I guess, unassuming PhD student who went on to become a professor and then eventually winning the Nobel Prize. I feel like we should say the title, Gene Machine by some Venki Ramakrishnan.
What?
Presumably. No, as in, we're not sure if that's the name, but we'll find it.
No, I am like 99%
okay.
sure.
Yeah, but the thing that really stood out for me about his story was how from the beginning he was very, or at least from how he told his own story, is that he put a lot of emphasis on like talking to other scientists.
Yeah.
Right?
Yeah.
It was always about trying to find out, I mean, on some level it's about competition, so you know what other people are working on, so I guess you don't overlap or whatever.
Yeah.
But also like talking to people about your problems and I guess asking for help. And
absolutely.
You know, I think that really like, you know it sort of fundamentally, but it never sunk in. But just reading of how much he did it made me feel like, okay, I am not even doing like 1% of this.
Yeah.
I should definitely reach out to more people.
Absolutely.
Talk to more people. And there was this one bit even where he wanted to use like some new instrument, and it's just him writing a cold email to somebody saying, hey, I want to use this, right? How often do I write cold emails? I've never done that, probably. Zero. So that's something I really want to like remember and like try to work on. The second thing that also stood out from the book for me was I feel like he was not somebody who shied away from like switching topics, right? Because early on, if you read it, the book is a lot about how he got started with something, but him realizing that this is not the problem that he wants to work on, and he moved multiple times until he finally found the one thing and presumably stuck with it for so long, right? So
that's a good one.
Yeah, that was also interesting because I do feel like I probably have some inertia if I just even think about changing topics or changing fields, especially like if it's a big change.
For me, actually, I feel excited to change fields in a way. I haven't changed it like very majorly yet.
Yeah.
Of course, there's the issue of you not being the expert, and you will have to learn a lot, and you might not have the competitive advantage of now being in this new field. But I'm not scared of it, and I'm quite excited about it as well.
I see. Well, for me, it sounds exciting. Like we talked about me wanting to do neuroscience at some point, right? It sounds exciting, but what I'm saying is like I've never taken any steps or like I've never done anything tangible to make that change. It's been more like, and then by change I mean deliberate change. For me, it's more like whenever something comes to its natural conclusion, then I move on to the next thing. And if there's
I mean, that is how it would be, right? Why would you, if you are doing something exciting or like you're doing something valuable or whatever, why would you want to do something completely different at that point in time? It's mostly like when things go down, or
unhappy
it is,
or something,
or like it needs this other push, or it needs like, or you talk to someone again. I think that is also where really all of these things come in, where you're like, oh, this guy doing completely different things are really exciting. Maybe I could help him with this blah part, and then you see how that goes. Of course, I have not done a lot of it, but of course, from wet lab to dry lab and like sort of the research that I am doing now is still cancer, but still different.
Yeah.
So I do like that component of active learning. I like that component of now talking to people of different mindset than what you were doing before, and now being able to know things from both ends, and then seeing how they connect, and then seeing how they should be talking. And I think that's pretty cool. And I think why I'm saying or like emphasizing more on this now is because I read 80,000 Hours again, and there they talked about how research, of course, is one of the most impactful fields if you do well.
Yeah.
And even in research, there were some problems which seem to be like more useful for the world. One being like studying pathogens or studying infectious diseases, which affect so many underdeveloped countries. So, I mean, at that time, I was obviously like, oh, fuck cancer.
Yeah.
Who cares about cancer anymore? Like, of course, I am doing it for now because I have been doing it. And that was a point in the book as well, where they're like, now making active decisions. Like, you have been doing things as they came to you, or you fell into them and you kept doing them. But sometimes you do need to make active decisions in terms of, oh, okay, I want to do this other thing now.
Yeah.
Even if all of this I'm doing is nice and cool and fun. I'm not saying I'm gonna do infectious disease right now. It's not so easy, but I am not scared of it either. Like, I think I would be excited if I find right people, if I find like right conditions and things like that.
Yeah, I guess then it's worthwhile, I guess, to keep your horizons open. Right? If you find something, then you switch.
Yeah. I think it's also about like, I know people who are very risk averse in a way. Like I know people who really just want so much stability in what they're doing and are scared to change any of it. In general, that stops people from expanding.
Yeah.
You would not know until you try. It might fail. Obviously, there's a high chance it would fail, but it might work also.
Yeah, I think I don't have that with the questions I pursue, because obviously I'm doing chip and bacteria. So this is the definition of risk.
Yeah.
But I think it might be true in a more like broader life sense, in the sense of life, right? Like as in the jobs you take. I feel like maybe maybe a lot of people are being risk averse in that domain of like, if let's say you are somebody who's a PI, now you're thinking about funding, and probably some topics are like easier to get funded, right? And in that domain, perhaps you are risk averse. And that feels very different from...
I did not think from that point of view. Yeah, that's a completely different ballgame.
Yeah. Anyway.
Yeah.
Yeah, any other takeaways from the book? Because apart from these two, I actually thought the book was a bit overrated. That was my takeaway. Yeah.
I mean, for me, I think, the 2 points you mentioned were definitely, they also spoke to me. And I think I left it off after the point where the crystallography took... like it was there for like pages and pages and pages, and I was just like bored of that part. But his excitement was infectious. His excitement was just like, he's just looking for something. He's like just so, but just the fact that he was so excited to do this bit, just this machine. He moved countries. He took his family, and like those big steps just for his curiosity and his excitement. That was kind of like quite commendable. And I guess like you need some of that, maybe not to that level necessarily, but to reach somewhere, or like, to achieve something big, that's that huge drive and just excitement, I guess. Yeah,
that's interesting you say that, because like Sue was the other person who recommended this, and she also said like that basically that the book was like a roller coaster, and she was like hooked from the beginning. But my experience of reading it was that I felt he's not actually a very good storyteller. In my opinion, like he gets a bit lost in the details sometimes, and I get his point of view. He doesn't want to leave out contributions by other scientists, right? Which might, I guess he's being considerate and trying to like show everything that really happened in a linear fashion. But that's not a great storytelling strategy, I feel like.
But again, it's a biography. Like if you write your own thing,
you can talk about you can give detail, but I feel like if you're writing a novel, you still want to weave that into like a nice narrative, right?
I mean, I think your standards are higher in terms of
my
getting
standards are higher.
Being like, you write yourself as well, right? Maybe you look at writing in a more defined, more
so the reason I got through the book was a bit I don't know if that was the same issue for you, but like there were bits where that got too much too descriptive, like
that did have
talking about very very like incremental changes. I get that that's the nature of the work, but you can like summarize it, right?
Agreed. I mean, that is why I think in a way I lost.
So how I got through that was I just skimmed through it. It's like, okay, done.
I did do that as well, but maybe I should get back to it. Like sometimes it happens, like you lose it and then you just lose track of it.
And there are some chapters I skipped completely. Like I did not care much for the Nobel Prize politics. Who cares?
I mean, of course, his life would have been affected at that point in time, but But the other thing I was gonna say is I liked how I got to learn biology as well, like very fundamental things, and the way like he is the pioneer, right? Like or him and his team, obviously, about these ribosomes and like the smallest. Of course, like again, it was boring when they would talk about one protein of that ribosome. Obviously, it's important, but like he's like, okay, we use this technique, we found these many. Now we use this technique and we found these many. That was getting repetitive, but I enjoyed like how the biology was being discovered with time, with all the small things that he was changing, and now like the very fundamental questions that were there in his mind and how he went about it was exciting to me.
Sure. True. Like how the discoveries were made, that was interesting. I would also say the same. And maybe this is also because where we are, to a general reader, I don't know how difficult.
I think it's quite technical. Like not super technical, but if I lost interest being a biologist.
Forget about the incremental stuff. So there are also bits when he's explaining the actual biology, right? Like how genes work or like how ribosomes work. I also felt even that was not a great description.
I see.
Yeah, and but maybe because we know how that works, maybe that's why I was a bit bored. But I feel like I'm contrasting this to like Richard Feynman explaining physics, for example, right? It's just like anybody can, like, it's so effortless to pick it up.
Yeah.
I did not get
But I guess maybe
kick.
like the same thing, that these concepts of physics, some of them maybe you were not knowing. You did not know.
Yeah, that's
so
why
now
I
him
wonder
telling, it's more exciting.
how somebody does not know would read it.
Possible.
Yeah.
Possible. Yeah, but Feynman is really cool.
Yeah. Yeah, okay, now that I shit on it, I feel like I should end with a positive note. Yeah, I still feel like I learned a lot. I would still, like, that was worth reading for sure, given where we are as scientists.
Yeah. And also, like, we talked about him giving the importance of connections and talking to people.
Yes.
But also the environment. That obviously includes people, but so many times he emphasized on LMB, the lab of molecular biology.
Seemed like a good scene.
Which was right next to our lab.
Oh,
my friend is from there. I don't know if the culture is still the same, but I could see that the people were so collaborative and like everybody's talking to everybody. The meeting rooms are like all kind of open, like glass windows. There's the canteen that he talks about where they would go up and discuss and like just hang out and have tea. That's very crucial.
Let's give him some slack. He did win the Nobel Prize after all.